Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter of class 12, that is molecular basis of inheritance. Dear student, in this chapter, we have already completed with the structural model of DNA, different experiments which was done for the discovery of DNA as a genetic material, the process of replication and their experiment. In transcription also we have discussed about the transcription unit, then different types of RNA, then we can say the prokaryotic transcription and eukaryotic transcription. But today in this lecture, we are going to talk about the translation. And as you know, translation is the protein synthesis, where the amino acid form a polypeptide, no polypeptide, amino acid form a polymer known as polypeptide. Polypeptide. Okay, so that polypeptide will again combine with another polypeptide to form a functional protein, or that polypeptide can act as a protein itself. So translation is the refers to the process of polymerization of amino acid to form a polypeptide. And you know that the amino acid are going to be added according to the genetic code which is present on mRNA. So whatever the genetic code on the mRNA, the sequence of genetic code on the mRNA will define which amino acid will be added in a sequence. So the order and sequence of amino acids are defined by the sequence of bases in the mRNA. So as you can see here, this is the mRNA. So which three nucleotide form a codon? That codon will specify that which amino acid is going to be added. And same with amino acid keep on adding, and between the two amino acids there is a formation of peptide bond. And due to the formation of peptide bond, the chain of amino acid is going to be formed, which will be known as polypeptide. The order and sequence of amino acid are defined by the sequence of bases in the mRNA. Dear student, this is the whole structure which we will see that this is the peptide bond between the two amino acid, and here this is the whole growing polypeptide chain. This is a peptidyl transferase center, means whole this polypeptide growing polypeptide will be transferred from here to here. This is tRNA. This is large subunit of ribosome. This is small subunit of ribosome. This is mRNA. In such a way, the whole complex is getting formed, where according to the codon on the mRNA, the tRNA, Charles tRNA will come, they will come with the particular amino acid according to the codon on the mRNA, and the amino acid are keep on adding. How they are getting added, how the process goes on, that we will discuss in the later part. The amino acid joined by a bond which is known as peptide bond. Okay. The bond which is formed between the two amino acid, that is peptide bond. So formation of peptide bond requires energy. It is an energy driven process. Okay. Now here, if we talk, the main machinery which is responsible for the synthesizing protein is nothing but a ribosome. And as we know, ribosome consists of structural RNAs, means ribosomal RNAs, and different proteins. Okay, so that is seen. So the cellular factor responsible for synthesizing protein is the ribosome. The ribosome consists of structural RNA, that is rRNAs, and 80 different proteins. As you can see this structure, this is the large subunit of ribosome and this is a small subunit of ribosome, and in between this mRNA is present, and this is the tRNA, and over which the growing polypeptide chain is added. It is a somewhat schematic diagram look like this. Means as you can see the polypeptide is forming, the two subunits are joined with each other. So in its inactive state, it exists as a two subunit, a large subunit and a small subunit. When the small subunit encounters the mRNA, the process of translation of mRNA to protein begins. Means, dear students, one thing you should remember that in cytoplasm, this synthesis is going to occur. These ribosomal subunits are separate from each other. They are not joined with each other. But when they start protein synthesis or they particularly carry out photosynthesis, during which these both subunits bind with each other. That is a large and small subunit. Okay, that bind with each other. And when small subunit particularly encounter the mRNA or bind with the mRNA, the process of translation begins. There are two sites in the large subunit of subsequent amino acid to bind and thus be close enough to each other for the formation of a peptide bond. As you can see, two sites in the large subunit for the subsequent coming amino acids to bind to and thus be close enough to each other. Now, if we talk about large, particularly, this is a start codon, this is the mRNA, this is a small subunit mRNA complex will form, and this is known as translation initiation complex, that is by the small subunit and tRNA and the large subunit. Okay, when this and here when tRNA join, large subunit or tRNA joins or initiate, it is a GTP driven process. We'll discuss that. And the ribosome also acts as a catalyst because 23S rRNA in bacteria is the enzyme or ribozyme. rRNA acting as enzyme because it's involved in peptide bond formation. And if we talk the translation unit in mRNA is the sequence of RNA which is flanked by a start codon and a stop codon and codes for a polypeptide. That is a translation unit. So suppose as you can see, this is the whole codon of mRNA. So AUG is start codon and UAG is stop codon. So this particularly a whole we can say a translation unit and that will code for our amino acid sequence. So this is the RNA sequence and this whole is nothing but considered as translation unit on mRNA, which is going to form a amino acid. And an mRNA also has some additional sequences that are not translated and that are referred as untranslated region and where they are located that we have already discussed in the mRNA part in the previous lecture. And we know UTRs are present in the both 5' and before start codon and 3' and after start codon and they provide efficiency they required for the efficient translation process. As you can see, this is the coding sequence, and before this, there is a UTR, and before that cap is there. 5' cap is so UTR is present between the 5' cap and before start codon. So after stop codon, again UTR is there at 3' and then poly A tail is located. So mechanism of translation and mechanism of translation, if we talk, then first we need to draw the structure of RNA, structure of ribosome. If we talk about structure of ribosome, dear student, then there is a large subunit and small subunit. If we talk about a ribosome, then it can be 70S ribosome, that is a prokaryotic, and 80S ribosome, that is eukaryotic. If it is a 70S ribosome, it has a large subunit and it is a small subunit. Large subunit is 50S and small subunit is 30S. Now, in between these two, three sites are there. One is E site, one is P site, and one is A site. Same way, in ATS also, one is large subunit and one is small subunit. Large subunit is 60S and small subunit is 40S. EPA. Now, here EPA, if we talk, the E site, P site, and A site. We remember E site stands for exit site. For whom? For tRNA. Means tRNA going to be exit from this part. Then P site is peptidyl transferase site. Means peptide is going to be transferred. Okay. And the growing polypeptide chain is associated with the tRNA which is present at the P site. And A site stands for acceptor site for charged tRNA. Like this whole process occur. Okay. And we remember this large or small subunit anyhow they are made up of rRNA and protein. We know that in 50S subunit there is a 23S rRNA is present and 5S rRNA is present. These are present in a large subunit of prokaryotic ribosome. And 16S rRNA is present in a 30S subunit of prokaryotic unit. Here also 20S 8S rRNA, 5S rRNA, and 5.8S rRNA are there, whereas 18S rRNA is here. And in this, if we talk about the P site, this P site is common for both large and small subunits. So if we talk E and A site, now dear students, this E and A site are of large subunit. This E and A site are of large subunit, they are associated with the large subunit. But P site, it is common for large and small subunit. Okay, in such a way the ribosome structure are there. Now we are going to discuss the process of translation, and when we talk about the mechanism of translation or the steps of translation mechanism, when we talk about the steps of translation mechanism, the first step we are going to discuss, that is activation of amino acid. And when we talk about activation of amino acid, it is amino acid getting activated in the presence of enzyme. So in the presence of enzyme, amino acyl tRNA synthetase, amino acyl tRNA synthetase, okay, that is enzyme, specific amino acid bind with ATP. Means by giving ATP, specific amino acid are going to be activated. So there are different amino acid. If we are taking any one amino acid and we are making it to react with the ATP in the presence of enzyme, that is amino acyl tRNA synthetase, and magnesium, the AMP get added with the enzyme and amino acyl AMP even complexes form and pyrophosphate get released. So it is a amino acid AMP even complex and it is nothing but a activated amino acid. Now after the activation of amino acid, the charging of tRNA take place. So that is the second step in this, that is known as charging of tRNA. And charging of tRNA, the specific amino acid are added or transferred to the tRNA. So here, what happens? The amino acid AMP even complex react with specific tRNA. Means for separate amino acid, the tRNA are specific. So if there are 61 different codon are there, 61 different types of amino acids are possible. Okay. And according to the codon, different types of amino acid are there. So this tRNA are also specific. And here, when react with specific tRNA, amino acid get transferred to tRNA. As a result, as a result, enzyme and AMP are liberated. Okay. So amino acid, our activated amino acid are getting added to the tRNA. Because of this charging of tRNA is also called as amino acylation of tRNA. Okay, so here activated amino acid is like this, and it is getting react with the specific tRNA. So we are naming it a tRNA1 because it is the amino acid 1. So now this, the amino acid get added to the tRNA 1. AMP, an enzyme get released, and this is nothing but a charged tRNA or amino acylated tRNA. So if you draw a drawing of this charged tRNA, it is suppose like this somewhat. This structure of tRNA we have studied. It is looking like this. So based on the mRNA, suppose this is mRNA. So if mRNA have AUG, then anticodon will have a UAC, and here the tRNA will have a charged tRNA, means it has a minus, so that is methionine. So if mRNA has a AUG, then the AUG codon, the anticodon on the tRNA will be UAC, and UAC containing anticodon containing tRNA will have a methionine at the 3' end. And like this tRNA will take this activated amino acid, this methionine, and come to the ribosome, and the addition of the particular amino acid occur. So now this way the process starts. Now later on, this amino acid all are activated and charged, then the formation of polypeptide chain initiates. So, therefore, in the first phase, amino acids are activated in the presence of adenine and linked to their cognate or their respective a process commonly called charging of tRNA or amino acid elevation of tRNA, to be more specific. And here you can understand it diagrammatically that this is a amino acid tRNA synthesis enzyme. This is a site for the amino acid, and this is a active site. This is a tRNA synthesis enzyme, and this is the tRNA. So tRNA bind with here. This is the particularly charged tRNA, and here you will see the particularly active site where a reaction occur. So this is the tRNA, this is the mRNA. mRNA have a codon. According to this, anticodon will be there, and the codon code for a specific amino acid. That type of amino acid with the anticodon to this codon will have this specific type of amino acid at this 3' end. Okay, so amino acid tRNA amino acid bond is formed, which is a high energy bond. Okay, that you should remember. So this way, all particular tRNA with their specific what amino acid they bind. And for initiation, the ribosome bind to the mRNA. The first step out of one, the ribosome bind to the mRNA at the start codon that is recognized only by the initiator tRNA. So first thing which is going to be take place, that first this small subunit is going to bind with the mRNA, and mRNA have a specific codon. This codon will lie at a P site, and the particular tRNA will bind here. Then large subunit of this particular will bind. Okay, so codon anticodon bonding will be occur. Now we are going to understand this formation of polypeptide chain in detail. Formation of polypeptide chain. Now this formation of polypeptide chain, it completes in 3 steps. It completes in 3 steps. First is chain elongation, chain initiation. If you want to form a polypeptide chain, then first polypeptide chain formation initiates, and this is known as chain initiation. And in chain initiation, first thing which happens that mRNA bind with small subunit of ribosome. Now you know that small subunit of ribosome are 30S or 40S. So if we are talking mainly about the 30S, in case of prokaryote, then in prokaryote, 30S subunit of ribosome bind with mRNA and it form 30S mRNA complex. And in eukaryote, 40S subunit of ribosome bind, which is a small subunit, bind with the mRNA and form 40S mRNA complex. 40S mRNA complex. So 30S mRNA complex will form, and 40S mRNA complex will form in eukaryote and prokaryote respectively. Now, how this complex look like? Withdraw. Withdraw. Now, dear students, be remember I am continuing with the prokaryote and what will happen in eukaryote that I will discuss. So in prokaryote, if you see, this is the mRNA, this is the particularly small subunit of ribosome, and here AUG. So AUG will come and establish at the P site of this small subunit of ribosome. This is a 30S subunit, and this is the mRNA. So mRNA with AUG codon stabilized at the P site of the small subunit of ribosome, and P R P subunit have a 16S rRNA. On the 3' end, some sequences are there that are going to bind with the sequences which are present in front of the AUG, and due to which the mRNA gets stabilized over the small subunit ribosome. If this bonding does not occur, for temporary bonding does not occur, what will happen? The mRNA will slip off. It is not going to be remain attached to with this one. Now after this, what will happen? First tRNA. So it is a what? It is, we can say, this is nothing but 30S mRNA complex. It is going to look like this. Now after this 30S mRNA complex, to it, the first tRNA is going to come and bind. So next what is going to happen? 30S mRNA complex bind with specific charged tRNA. Okay, so if it is going to bind with the specific charged tRNA, the initiator codon is there. So this charged tRNA have methionine amino acid. So here, this is the small subunit of ribosome. This is mRNA. This is AUG. Okay, AUG codon is there. As we have discussed, the 16S RNA form temporary bonds, and to this AUG, UAC will be the anticodon, and UAC anticodon containing tRNA. I'm just writing a name tRNA, so you should understand every time there is no need to draw a similar structure, but you can remember the structure which we have studied. So tRNA is there, and tRNA will come here with the specific amino acid, that is methionine. Okay, so this is 30S subunit, and this 37 subunit with the P site, and at P site the codon anticodon bonding occur, and the first charge tRNA is going to edit. Now be remember one of the important point, that in prokaryote, non-formilated methionine is attached to tRNA, and in eukaryote, formilated methionine is attached to tRNA. So here it is just formilated methionine is not there, but if it is a 40S subunit, here tRNA, FMET tRNA Formaldehyde, methionine, and tRNA will going to bind. Then after this, the large subunit is going to be added. See, I am drawing the diagram of only prokaryotic. Similar diagram will happen to the eukaryote. Now in eukaryote, similar mechanism is happening. Just only the factors are different. So after this, what happens in chain initiation? The larger subunit bind with 30S mRNA charged tRNA complex. So here the diagram will look like this somewhat. This is the mRNA. This is a small subunit, that is a 30S subunit. This is a P site. This is AUG. The anticodon is UAC. tRNA is there. tRNA have a methionine, and the large subunit is going to be added. So this is the 50S subunit, and this is the particularly 50S subunit in eukaryote is getting added, and 60S subunit in eukaryote getting added, and the small subunit, large subunit mRNA charged tRNA complex is going to be formed. So, and this is the initiation complex that is formed. Now after the chain elongation will occur. Now in chain elongation, what we need to read whatever the codon are given after this AUG. Suppose here UUU is given, UCG is given, CAC is given, UCG is given. Okay, like this it is given, and later on UAA is given. Okay, like this it is the sequence in mRNA from 5' to 3'. Now we are going to understand the chain elongation. So chain initiation up to the binding of large subunit. Now after this, second we are going to talk about, that is chain elongation. Chain initiation is complete. In chain elongation, what is our target? Our target is to read the mRNA strand, a codon on mRNA strand, and add according to specific their amino acid and make a polypeptide. So here it is known that after formation of complete ribosome, mRNA, tRNA complex, A site is established next to P site. Why? Because A site is a part of large subunit. So when these large subunits are binded, it is established A site and E site here. Okay. So A site is established next to the P site. And due to the establishment of A site, it exposes mRNA codon next to the initiation codon. Okay. And if next codon, suppose as I have written that it is UUU after AUG, then UUU is getting exposed. So according to UUU, a new amino acyl tRNA complex reaches the A site and form codon anticodon bonding. Now, as we have drawn in the previous diagram, the second codon is UUU. It can be anything in different mRNA. It is going to be different, but the initial codon is going to be AUG. So first amino acid is going to added with the tRNA, that is methionine in a polypeptide. But at the A site, when large subunit come and bind, A site is getting estabilized. If UUE is getting exposed in any another mRNA, another amino acid is going to be exposed because it is going to be varying. So if it is UUU, the phenylalanine containing tRNA is going to come and bind and reach to the A site and form anticodon codon anticodon bonding. So now when codon anticodon bonding, this require elongation factor and energy, that is GTP. Okay, so now how it look like this? This is the mRNA. This is the 5 dash. This is the 3 dash. Here it is small subunit is binding like this. This is AUG, UAC anticodon containing tRNA is here with methionine amino acid. But at the second position, UUU come and establish at the A site. So UUU, the anticodon is AAA. Then here another tRNA will come, and this tRNA with the AA anticodon has a phenylalanine amino acid because UUU code for phenylalanine according to the triplet codon. Now, then large subunit is going to be large subunit is already binded, and at A side this charged tRNA is getting added. Now, after the addition of this charged tRNA, next is what going to happen. Next, you will see that a bond, this methionine, is shifted to here over the phenylalanine. Okay, this methionine is shifted over the phenylalanine and a peptide bond is formed. So, amino acid on charged tRNA at P site is shifted on amino acid of A site and peptide bond is formed between COOH group of first amino acid, that is methionine, and NH2 group of second amino acid. Like this, two charged tRNA are close enough and the peptide bond is formed by shifting here. Okay. So now next diagram is going to be look like this. and methionine and phenylalanine like this attached. Okay. So now this two charge between the two T amino acid, the peptide bond is formed and which is energetically favored. And this here, this peptide from peptide or amino acid, we can say. Later on it will be. So peptide or amino acid transferred to amino acid on A site by 23S rRNA. Here 23S rRNA act as catalyst and show peptidyl transferase activity. So peptide bond formation is enhanced. And after forming a peptide bond with the amino acid at the A site, and it is favored by the 23 sRNA because it is going to be particularly in case of prokaryote. Now, same mechanism is done by 28S rRNA in eukaryote. Okay, 28 rRNA in prokaryote and 20S rRNA in eukaryote carry out same reaction. Okay, but in prokaryote 23S RNA is involved in formation of the particular peptide bond. The elongation factors are required in this process. Now after this, what will happen? After this, the translocation will happen. So in translocation, what is going to happen? In translocation, this tRNA on the A site is going to be shifted to the E site. P site is going to be shifted on the E site, and whatever the content which is present on this particular A site is going to shifted on the particular this P site. So next, what will happen? Next, the translocation occur where movement of ribosome take place on mRNA. Okay, on mRNA, ribosome movement take place, and ribosome move from codon to codon along the mRNA. Now, if suppose it is moving, now what is going to happen? The AUG, if ribosome shift over the mRNA, AUG will initially it was present at the P site. Now it will come to the E site. At P site, UUU will come with the particularly that having what AAA anticodon with its specific tRNA with phenylalanine and methionine peptide. And now large subunit is there where also the A site is now free. The tRNA from the E site going to be exit, so it get exit, but A site is now free for entering of the another amino acid. Now suppose UCG codon on the mRNA is getting exposed. Now UCG, if we talk this UCG code for serine. So now UCG having an anticodon AGC and AGC anticodon tRNA with serine will come and bind here. Now when serine will added here on this tRNA, now next the further steps are same. What this peptide is going to be transferred. So after this what happened? Peptidyl transferase activity by 23S RNA. And due to this peptide transferase activity, now the sorry dear students, this is methionine. Okay, now this peptide is going to be shifted to on the A site. Now after it is shifted on the A site, again the bond will form. After bond will formation, again the translocation will happen. So peptide transferase activity will occur on the 23S RNA. And if peptide transferase activity will occur now on the particularly this tRNA will remain here, but here it is a nothing but a for serine we can say UCG. Now if it is UCG, AGC will be there, tRNA is here. Now tRNA with the serine, but bond is formed between the phenylalanine and serine and the methionine. Like this peptide is going to be formed. Now after this elongation, sorry, translocation, then again A site codon get exposed, exposed, and now repetition of filling of A site with charged tRNA, peptidyl transferase activity, and translocation till stop codon reach to A site. So now stop codon will reach to A site and chain termination will occur. In such a way, the whole mRNA sequence get translated into the polypeptide sequence. Now the chain termination will occur. Now in chain termination, now the polypeptide which is formed is going to be released, or polypeptide synthesis is going to be terminated. So here termination of polypeptide is signaled by one of the three termination or nonsense codon, which are UAA, UAA, UAG, and UGA. Now, this stop codon do not code for any amino acid. So, any amino acid, when stop codon will come at the A site, is not going to be added by any tRNA. Okay. So, amino acid are not coded by stop codon. So, GTP dependent factor known as release factor associate with associate or bind with termination codon. Now we remember in eukaryotic release factor 1, only one is used, only one release factor is there. But in prokaryote, actually three type of release factor 1 are there: RF1, RF2, and RF3 type of release factor are found. And this release factor bind with the stop codon and release the complete polypeptide from the ribosome. So now how it will look like this? Suppose this is mRNA. Now ribosome has reached the stop codon. Suppose UAA, and this is a 3' end. Before any amino acid was there. Suppose CC, and before that GCG. Any amino acid was there. Now small subunit is here, large subunit is here. Okay, the particularly specific tRNA is here. And with this a long polypeptide is also there, which is based on the reading of this codon. tRNA is getting exited, and here new codon is charged tRNA is not going to edit, so release factor will bind. And when release factor will bind, translation get terminated and complete polypeptide is released from ribosome. Such a way, whole polypeptide is getting my mRNA is getting read, and the amino acid are getting added, and at last the complete polypeptide is going to be released such a way. So this is the complete polypeptide, which is going to gain its conformational change and gain a proper structure, gain stable structure, and that is regarded as a protein. Okay, so like this, the whole translation process occur. Now this is the process. Now we remember in prokaryote and eukaryote, different type of factors are used, like initiation factor in initiation, during elongation, elongation factor are used, and during termination, termination factor are used. They are different in eukaryote and prokaryote. There are different types, but we have not talked about that factors because that much detailing is not asked. But sometimes the question about one or two factor is getting asked. So what is the information required for that? That I am making you to write here. That will help you to go solve the question. So we remember some important point that in chain initiation or during chain initiation, three initiation factor in prokaryotes and nine initiation factor in eukaryotes are required. Okay. After this, we remember one GTP is used. Sorry, first one ATP is used during activation of one amino acid, and one GTP is used when one ATP is used when Charles tRNA reach to A site. Okay, and codon anticodon bonding occur, one GTP is used at every time when a new Charles tRNA reaches the A site. And next, one GTP. Now we remember when Charles tRNA or new Charles tRNA reaches to A site, it requires elongation factor. It requires elongation factor. That is eukaryotic elongation factor 1 in eukaryotes and elongation factor Tu and elongation factor Ts in prokaryotes. Now the question is, what is EFTU and what is EFTS? Now EFTU is elongation, stands for elongation factor thermo unstable, and EFTS, that is elongation factor thermostable. Means EFTS is temperature stable or at high temperature it is stable, whereas the EFTU is thermo unstable, means which can degrade. Now it has been found that Escherichia coli has its most abundant protein, that is EFTU. Okay, like this is particularly used. And next, one more GTP is used during translocation, and here elongation factor gene, which is a elongation factor in prokaryote is used. So here you can see that one GTP here. In the previous slide, we have seen that one more GTP is getting used and one ATP is getting used. So for every amino acid getting added, for every new amino acid to get added, one ATP and two GTP are used. Okay, or we can say for every peptide bond formation, this one ATP and two GTP are used. So if we talk here, then this is all about what a particular translation thing. Now we are going to deal with one question. And if we deal with one question, the question is, how many ATP and GTP molecules are required respectively for incorporation of 25 amino acid in peptide chain. Okay, now we know that first we should go with the options, and given options are A is 20 ATP and 20 GTP, B is 25 ATP and 25 GTP, C is 50 ATP and 50 GTP, and D is 25 ATP and 50 GTP. Now, dear students, in just previous slide, we have studied that for addition of one amino acid, one ATP and two GTP are used. So here, how many? 25 amino acids. So 25 ATP and into 2 GTP for each amino acid. So 50 GTP. So option D is going to be the correct answer. Okay. So that is all about what particular this. Now, after this, at last, we are going to discuss some of the antibiotic and their effects. Okay. Now, we are going to talk about some antibiotics and their effect. The first antibiotic is streptomycin. It is known that streptomycin inhibit initiation of translation and causes misreading. Okay, actually, how this antibiotic can help? Suppose we take a petri dish and in this we add a particular antibiotic, and bacteria is not going to grow. Why it is not going? Because it is inhibiting some of the protein synthesis process for any particular target site. So here streptomycin is going to inhibit this protein synthesis by the inhibiting the translation. Okay, by causing a misreading. Second is tetramycin, and it is known that tetramycin inhibit binding of amino acyl tRNA to ribosome. In such a way, it inhibits translation. Third is chloramphenicol. Chloramphenicol inhibit peptidyl transferase activity by inhibiting the peptidyl transferase enzyme, and so peptide bond formation. Now fourth is now fourth antibiotic is erythromycin. Erythromycin inhibit translocation of mRNA along ribosome. Then fifth is neomycin. Neomycin inhibit interaction of tRNA with mRNA, means codon anticodon binding. Sixth is puromycin. This binds with C terminus of growing polypeptide, or a free COOH group in the growing polypeptide chain, and cause premature termination. Premature termination of protein polypeptide chain in both prokaryotes and eukaryotes. And sixth antibiotic are rifampicin and actinomycin. Actinomycin. If we talk about this antibiotic, they inhibit RNA synthesis by inhibiting RNA polymerase. You know, students, that if RNA polymerase will not occur, RNA synthesis will not occur, and without formation of mRNA, translation cannot occur. So this way, this antibiotic work on the translation and some of the site of transcription, and the particular translation is going to be inhibited. So amino acid are added one by one, translated into polypeptide sequence, dictated by DNA and represented by mRNA. Be represented, dictated by mRNA, but actually represented by mRNA, because here mRNA is getting used. And at the end, we have studied that release factor bind to the stop codon and terminate the translation and releasing complete polypeptide from the ribosome. Okay, you can see this particular process, how the particularly whole process we have discussed that occur, how the new charged tRNA come bind with this, then peptide bond formation occur, and this process going on. Okay, so this is the whole process which we are going to see in this one. See translation process, how it occurred.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into three stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail.
Please understand, this is just revision.
mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basics.
So this way, I hope you understood this particular one, that how this translation occur. Okay, in this 3D video manner, let's discuss some of the MCQs. Process of polymerization of amino acid to form a polypeptide is called translation. You are no better way. Now you are clear with this. Now amino acids are joined by a bond which is known as, obviously, it is a peptide bond between the two amino acids. Okay, I hope you understood this mechanism of translation in detail very well. Thank you.